Hello, I'm very happy to introduce our new work, Biomedical Named Entity Recognition via Dictionary-Based Synonym Generalization. I am Zhao Fu from University of Cambridge. This work is collaborated with Yixuan Su from Cohere, Zaiqiao Meng from University of Glasgow, and Nigel Collier from University of Cambridge. In today's talk, I will first give a brief introduction of the biomedical named entity recognition task. Then I will show our newly proposed synonym generalization framework. After that, we conduct a theoretical analysis of our newly proposed component to prove its effectiveness theoretically. After that, we will conduct some experiment to show the effectiveness of our framework and the correctness of our theoretical analysis. Biomedical named entity recognition aims at recognize named entities from given text. Here is an example. Given a sentence containing many biomedical named entities, we hope our model can extract this kind of named entities from that sentence. There are many methods proposed to solve this problem. The supervised method, which requires large-scale human-annotated data for training. The distant supervised method, this kind of method uses weakly annotated data based on an in-domain training corpus. The last one is the dictionary-based method. This method only trained with predefined dictionaries and do not need any human annotation. So in this paper, we focus on the third method, dictionary-based one. Dictionary-based approaches have many advantages. It do not need for human annotated data or any in-domain corpus. Therefore, it becomes a popular choice because we do not have many human effort or expert involved. However, there's many challenges in this kind of methods. Particularly, it suffers from the synonym generalization problem. This problem means when recognized entities, it's only limited to the entities that exist in the dictionary, and it cannot recognize synonyms outside of the dictionary. Some methods propose to use string-similar tool to recognize the synonyms. However, this is still not enough. In this paper, we make several contributions. First, we propose SynGen framework. It is a novel dictionary-based method for the biomedical NER task, and we propose to solve the synonym generalization problem. Secondly, we conduct extensive theoretical analysis to show the correctness and effectiveness of our proposed SynGen framework. Lastly, we conduct extensive experiment to validate the SynGen's effectiveness over a lot of benchmarks, and it shows our model outperforms many existing dictionary-based models. Now, let's move on to the synonym generalization framework. This is an overview of our synonym generalization framework. It contains two stages, the training stage and the inference stage. In the training stage, it samples synonyms and learns to classify positive samples from negative samples. In the inference stage, it will first split the sentence into spans and give score to each span. Only spans with higher score will be recognized as named entities. Now, let's give a rigorous definition of the task. First, we denote the biomedical domain as capital D, and then we denote all possible biomedical entities in domain D as a set capital S. After this, we can define the task as identifying subspans in a text X that belongs to S. It should be noted that in the real world, we only have access to a subset of S. We call it S hat, which we also name it as a dictionary. In the training stage, we will first sample synonyms as positive samples and spans from biomedical corpus like PubMed, as negative samples. The objective of our model is to classify the samples using cross-entropy loops. We propose two novel regularizers, including synonym distance regularization and noise perturbation regularization. Let's first look at the dictionary-based loops. This is quite similar to previous methods. We first sample positive samples SI hat from the dictionary S hat, and then we will encode SI hat into an embedding RI hat. After that, we calculate the probability of SI hat being a span of entity with its embedding. Similarly, we calculate the probability of a negative sample NI tilde from PubMed with similar computation. After we have the probability for positive and negative samples, we can calculate the dictionary-based loops LC, which is shown as the last equation. To better sample negative samples, we propose the negative sampling filtering strategy. We will first sample randomness spans from the PubMed corpus, and then we remove samples that is too close to dictionary entities. Specifically, we will ensure that the distance between the embedding of SI hat and the NJ tilde, we ensure the distance is larger than a threshold TD. Now let's look at the synonym distance regularizer. The goal of SDR is to equip the model to identify more synonyms of the same biomedical concept. So if the embedding of the synonyms are very close to each other, the model are more likely to get a better result. Here is how we do SDR. First, we sample anchor entity SA hat and the synonym SP hat from the dictionary S hat. So the entities are synonyms of each other which share the same concept ID. Then we will sample a random negative span NN tilde from a corpus. And then we can impose a triplet margin loops as shown here. Here, gamma S is a predefined margin and R is the embedding of S. Now let's move on to the noise perturbation regularizer, NPR. The goal of NPR is to reduce the sharpness of the score functions landscape to give close entity similar scores. Therefore, synonyms of biomedical entities are expected to be close with each other. So the NPR is defined as follows. Here, RI hat is the embedding of the biomedical entity sampled from S hat, and V is a Gaussian noise vector. The aim of NPR is to flatten the landscape of the loss function, and it can minimize the loss difference for vectors with close regions. After we propose the two regularizer, we can combine them together with a weighted combination. Here, alpha and beta are tunable hyperparameters for the two regularizers. In the inference stage, we will first split the input text X into spans, and then we will give each span a score. After that, we only keep the spans with score larger than T. We will use the greedy extraction method to extract the biomedical terms. Here is how we do the greedy extraction. This is because we observe that in biomedical terms, such as T-cell leukemia, this biomedical entity can be nested and it contains many sub-entities like T-cell or leukemia. SynGen's greedy extraction component will first rank the recognized term by its length in descending order, and then it starts with the initial validation sequence X1 and set it to X. And then we will iterate for each item in the set T. If Ti is a subsequence of the current validation sequence Xi, we will first insert T as a biomedical entity, and then we will remove the subsequence in Xi, and we will do the iteration again. Now let's move on to the theoretical analysis. Most existing dictionary-based framework struggle with the synonym generalization problems, and we propose two components to resolve this problem. In this section, we hope we can give theoretical analysis of how and why our newly proposed regularizer can help to alleviate the problem. Now, let's move on to define the loss function and the average empirical error. The loss function is defined as the first equation. It is a bounded function between 0 and a positive number b. Then we can define the average empirical error. It represents the average of the loss function value across all entities in the dictionary S hat. So if R hat is low, the model performs well on dictionary terms. But a question arises, how about the performance on other biomedical entities outside of the dictionary? We can define the synonym generalization error to describe this problem. The synonym generalization error is defined as follows. Smaller ES means that the error for any S will be very close to R hat, which is quite good. However, if ES is high, it means that the model may struggle with many unseen synonyms. Before we conduct the analysis, we should make some assumptions first. The entity cover, we will assume that S hat is an epsilon net of S, which means for any entity in S, there is a close representative in the sampled dictionary S hat. So the distance between S hat and S will always smaller than epsilon. The next assumption is the flatness of loss function. We will assume that F is a kappa ellipsis function. So the loss function will not change too rapidly between 2 points. Now let's move on to the synonym generalization error bound. It is defined as follows. Given the above assumptions, with probability at least 1 minus delta, we will have ES is bounded by the right-hand side term. So it can be observed that there are a lot of terms that can control the right-hand term and then control the upper bound of ES. For example, the dictionary density epsilon, it refers to how tightly entities in S are clustered around those in S hat. So if epsilon is small, it will ensure a better generalization. The next one is the Lipschitz constant kappa. It represents the stability of the loss function. Therefore, smaller kappa will ensure that the function don't have a very big change, leading to the model's robustness. The next one is the dictionary size, the size of S hat. So larger size of S hat will help to reduce the error bound, which leading to the better generalization. It is very interesting to observe that if S hat size is very large, the second term of the right-hand side will be close to 0. So in this case, if we continue to increase the size of S hat, the performance will not increase anymore. Our experiment will also show this. The final one is all terms count, the size of S. So larger size of S will lead to bad generalization bound. Luckily, it only gets worse at the rate of O square root of log size of S. Now let's move on to the analysis of SDR. So we can observe that SDR aims to decrease the distance between the synonyms, and this reduction is very similar to minimizing epsilon. Therefore, it can diminish the synonym generalization error upper bound, and therefore it can help to control ES. The next, we will analyze NPR. So it can be observed that Lipschitz constant kappa is calculated as follows. Therefore, minimizing Rn is equivalent to minimizing the kappa. Therefore, minimizing Rn can also help to control the upper bound of ES. Now let's move on to the experiment part. We evaluate our model on 6 popular datasets, and we report the precision recall and the F1 scores for each experiment. We use the synonym dictionary from UMLS, and the hyperparameter is tuned with grid search. The negative sequence is sampled from PubMed corpus, and our backbone model is the PubMed BERT. We run all of our experiments on 1390 GPU. We compare our baseline model with many distantly supervised model and supervised model. The first one is BioBERT. It is first pre-trained an encoder with biomedical corpus and fine-tune the model on annotated NER dataset. The SBM is a standard span-based model for NER task, and the SBM cross uses the same setting with SBM. The only difference is SBM cross will test on another dataset within the same domain. The Shark is a weakly supervised method, and the AutoNER will train AutoFace and the tailored dictionary to do the NER. We also have many dictionary-based methods as our baseline. The embedding sim uses pre-trained model for the encoding, and the meta map do the exact concept mapping. SPED just use shortest path eated distance to extract the named entity, and the TF-IDF follows the method of UGS as the similar scores. We also compare with quick UMLS. It is a retrieval system to extract the similar entities. Here is the main result of our experiment. We can observe from the experiment that the SynGen framework outperforms other models in F1 scores, and we improve the recall by capturing more entities. By comparing with SBM and SBM cross, we can conclude that performance varies from the choice of in-domain corpus. If we made some wrong choice, it can decrease the performance. The SynGen's F1 score are very close to SBM cross. It shows its effectiveness in these tasks. Quick UMLS has a very high precision use exact match, but it will struggle with out-of-dictionary synonyms. Therefore, the score of recall for UMLS is very low. For AutoNER, we can conclude that the tailored dictionary is very important. Without such in-domain corpus, the performance will not be quite good. We also conduct many ablation studies to show the effectiveness of our framework. We can compare from the experiment that variant without NPR or SDR will drop in the performance. The NSF is also very crucial because it can help to improve the performance. The greedy extraction can also help to boost the precision. The next one, we can analyze the impact of the SDR component. So we change the alpha for different scores and run the experiment. We can observe that as alpha increases, the synonym distance will decrease, and the evaluation score will improve as the synonym distance is regularized. So we can make the conclusion that the SDR component can effectively control the synonym distance and boost the performance. The next one, we conduct the analysis of the influence of noise perturbation. We investigated the score change with different beta, and we can observe that as beta increase, all of the scores, including precision recall and F1 scores, all rise. Therefore, this observation also supports our theoretical analysis, and it shows our proposed component is very useful. The next experiment, we conduct the few-shot analysis. We can observe that the SynGen performance rises with the dictionary size, especially when the size is very small. We can use just 20% of the dictionary entries to get a very good result comparable to using the full dictionary. In contrast, the meta map performance only increased linearly with the dictionary size. The next experiment, we conduct the standard deviation analysis of our model. We run each experiment 10 times with different random seeds. And from the experiment, we can conclude that the SynGen framework constantly outperforms all the other models without the proposed component constantly and uniformly. Here is the conclusion of this paper. We propose a novel SynGen framework to conduct the biomedical NER task with only a dictionary. We propose 2 new regularizers to enhance the performance. We propose a very comprehensive theoretical analysis to show the effectiveness of our framework. We also conduct a lot of experiments to show the effectiveness of our framework and the correctness of our theory. Thank you very much.